So welcome everyone. This is one of our most enjoyable events of the year. It's a time we get to come together and really revel in outstanding science and engineering. So this is the DeLisi Award Lecture, and it's made possible by a generous gift by Charles DeLisi, who's Dean Emeritus of the College of Engineering and Metcalf Professor of Science and Engineering. As many know, but perhaps some don't, Charles is widely considered the father of the Human Genome Project. He was really an early pioneer in computational molecular biology. He made seminal contributions to theoretical and mathematical immunology. He held many distinguished positions, Lawrence Livermore National Lab, Department of Energy, DOE, and his most distinguished position was being Dean of College of Engineering here. And he really transformed us into a world-class College of Engineering. And after serving as dean for a decade, Charles then founded Boston University's bioinformatics program, which was the first program of its kind in the nation. So today, before we get started with the DeLisi Award, I want to honor the college's recipient of the Early Career Award in Research Excellence. So the award this year, it celebrates the extraordinary success in building high-impact research early in one's career. And the winner this year is Ashed Onbar. So Professor Onbar's research focuses on the intersection of artificial intelligence, computer vision, and robotics with applications in autonomy, accessibility, and sustainability. His work on inclusive design has resulted in high-impact projects such as OpenGuide, which is a human-like guidance system for visually impaired travelers. It's funded by the Department of Transportation and NSF. His NSF-funded work also includes a grant on inclusive mobile robots. His contributions to autonomous vehicle systems, including situational and robust learning algorithms, have defined benchmarks in the field. Through these projects and others, he has advanced the state of assistive technologies, enabling greater autonomy and mobility for individuals with disabilities. Professor Onbar has published extensively in premier venues, and his H-index is already 32, and he has over 3,800 citations. It's a testament to the far-reaching influence of his research. His work often bridges fundamental innovations and practical applications, addressing challenges in autonomous systems, human-computer interaction, and intelligent transportation. His work has garnered numerous accolades, including the prestigious IEEE Intelligent Transportation Systems Society Best Dissertation Award, a Humboldt Research Fellowship, multiple Best Paper Awards, and an NSF Career Award. Congratulations, Ashed. All right, and now we transition to the winner of the DeLisi Award. I am going to give some prepared remarks, and then our other Dean Emeritus, Ken Luchin, longtime friend and collaborator of Dr. Suki, is going to give some additional remarks. So Professor Suki was born in Hungary and graduated as a physicist from the University of Szeged, Hungary, where he received his PhD in biomechanics from the same university in 1987. As a postdoctoral fellow, he spent a year at INSERM and then a year in the Meekins Christie Laboratories at McGill University. Then he joined the Department of Biomedical Engineering at Boston University as a research associate, and then he became an assistant professor and a full professor in 2007. His current research focuses on the complexity of multi-scale structure-function relationships in the lung, ranging from subcellular dynamics all the way up to organ-level mechanics. His work includes the development of elastic networks to understand how collagen and elastin fiber rupture contributes to the progression of emphysema, how the characteristic structure develops in pulmonary fibrosis, and how dynamic stretch patterns affect cell function with implications for mechanical ventilation. He's an elected fellow of the American Institute for Medical and Biological Engineering, a fellow of the Biomedical Engineering Society, And he has received a presidential award by NIH to support exceptionally innovative, high-risk research projects that have the potential to create or overturn fundamental paradigms. He has been a visiting professor at multiple places around the world, and he's also an honorary professor at his alma mater in Hungary. He's co-authored over 260 peer-reviewed articles and 13 book chapters. In 2021, he wrote a book published by Elsevier entitled Structure and Function of the Extracellular Matrix, a Multi-Scale Quantitative Approach. This is a must-read for anybody working broadly in structure-function relationships. It's a true delight. And he is also co-owner of a startup company he founded, Mechanobiology, that seeks solutions to accelerate pulmonary drug development. In 2024, he received the Joseph R. Rodarte Award for Scientific Distinction from the American Thoracic Society. And now we will hear some remarks from Ken.
Well, fortunately, I just found out that there was going to be asked to give some remarks 3 to 4 minutes ago. So I'll try and keep these brief, but somewhat more personal and enlightening. Bela came to Boston University from his postdoc position. He was at the University of Szeged, as Elise informed us. He was with Jason Bates over at McGill University, and he came here as, I believe, a research assistant professor, and started his career here. It was pretty clear right from the beginning that we had a special person, someone that was astonishingly inquisitive, intellectually fearless in ways that I haven't experienced before. And I was an assistant professor at the time. He came out of Zoltan Hantos's lab, and what we found out is there was no aspect of the integration of science and engineering and technology related to lung function that Bela would not be able to impact and use. He has been an astonishingly productive individual at the use of mathematical and computational models, from the molecular mechanisms to the macro scale. Very few people can talk about a record of a scholarship at all those scales. He understands in the use and the application of fractals to the human physiology and, of course, the lung specifically. He had expertise in acoustics. He had expertise and got us involved in understanding nonlinear systems and how, because the lung is inherently nonlinear, and we always kept interacting with it and modeling and measurement-wise as if it was linear, as most engineers try to do, but it wasn't. And we figured out ways around that. Fluid mechanics at every scale. Estimation theory and estimation and parameter estimation and modeling and that. His expertise in signal processing and the application of random signals to probe lung function. These are seminal works. They're absolutely seminal works. One of the earliest things that we were trying to challenges we had came out of Zoltan Hantos's lab in understanding. Nobody was able to make measurements on the lung between about breathing frequencies, which is about 0.2 hertz, and about 4 hertz. Everybody was doing oscillations on the lung from 5 hertz on up, because you can then low-pass filter off the breathing signal. And his lab in Szeged has suggested there might be some really, really important information about lung disease down there. And we thought, well, how can we do this? How can we design a signal to do this? And this required very deep understanding of how nonlinearities and harmonics interacted. And Bela was essential in producing this thing called an optimal ventilator waveform for my research program that I was interested in. And we collaborated a lot on this. And this opened up a whole new understanding of what was going on in the lung and how heterogeneities in lung disease massively altered function. Bela and I have been to many, many scientific conferences together, and I lost count of the number of beers we've had in those conferences, but it's hundreds, if thousands, not thousands. Bela and I have together gone back to the University of Szeged, worked with Zoltan Hantos's lab. I've done that. We did it twice, I think, and it was a remarkable experience. And producing some really highly, I think my personal highest cited paper came out of that work. We also took some social trips to Budapest and others, and I can't possibly tell you anything about that without getting both of us in serious trouble. Although I do remember driving there and wondering, are you sure this is a two-lane road? Because you're on this other lane. Yeah, we have to pass this guy, but there's a car coming at us. I just want to just reinforce that I have been around a lot of scientists in my life, and I don't know anyone who is as robust and intellectually fearless and diverse in applying any technology, science, method I can think of to understanding better the lung function at every level. And I can tell you honestly, I am deeply indebted to Bela as a longtime colleague. My success is impossible to have happened without my collegiate relations and my collaborations with Bela. So thank you, Bela, personally, and thank you from the College of Engineering at Boston University.
Thank you very much. This is a wonderful introduction, and I am humbled, and I'm also honored for getting this award. I deeply appreciate it, and it's great to see a lot of friends, colleagues here, ex and current lab members, and actually when I learned that I would get this award, I immediately, enthusiastically started putting together the lecture. And my second attempt resulted in 55 slides. But that was a lot better than the first attempt, which was over 100. So anyway, I managed to cut it down significantly, so we'll see how it goes. But I guess the introduction was so generous, I could skip 10 of my slides. So these are the topics that I would like to talk about a little bit, just briefly about my career path, a little bit about my lab, and then 4 topics very briefly: biomechanics, fluctuations, modeling, and technology. Early life started in Hungary. This is my mother. Back in a typical Hungarian, Eastern European city. And then this is my father. They were both actually high school teachers. And my father was also a philosopher, and we always had some discussions. And anyway, he had a huge impact on my worldview as a teenager. And this is myself. I was about 4 years old, I think. And we lived in one of those apartment buildings right here. I don't know if you can see this, but somewhere like those. And I must have been around 11 when I found a box, a wooden box in the basement. So I opened it and I saw some electrical things in there, like resistors. I didn't know what it was. So fortunately, there was a guy, an electrical engineer in the apartment building. And so I talked to him. He said, well, this is a radio. He gave me a headphone. And indeed, I could hear something, but it was terrible. So then he said, well, let me put a transistor and a battery into it. So he did the circuit, and it was really great. And I was hooked. I started reading books on circuitry, and then later I migrated towards physics, and then I ended up as a physics undergraduate in the local university. After college graduation, this is Zoltán Hantos that Ken already mentioned, and this is actually one of those trips to Hungary, and here is Ken. He looked very nice. And Zoltán was a superb experimentalist, actually, and I learned a lot from him. And after this, I went to a laboratory in France. This is René Pellin's laboratory. He was one of the smartest and best respiratory physiologists of the time in Europe. And actually, this picture is really, really famous because when he died, the picture was put into one of the top respiratory journals. But the picture is actually also famous for another reason. This guy here, he's actually from Boston, and his name is Jerry Mead. He was from Harvard. He is the founding father of the field of respiratory physiology. After France, I joined Jason Bates's lab. And that was a wonderful year, almost a full year. And actually, ever since, we have stayed in contact. We published a lot together. And in fact, we just completed a book. That's the title. And Meanwhile, while I was there, Andy Jackson and Ken offered me a position at BU. And Charles DeLisi was kind enough to elevate the position to a senior postdoc level. And so I happily moved down. This actually came a much better salary as well. And so then I joined BU, where the fun started because of the opportunities that BU provided. And I have collaborated with many people from the department, among departments, and outside as well. And I just need to acknowledge a few. And of course, the first is Ken. We started collaborating early on, and essentially, he introduced me to the art of modeling as well as mechanical ventilation. We published, I actually counted, more than 60 papers together, and it's still going on. And so then, in the mid-90s, I got to know Gene Stanley, a very famous physicist in statistical physics. And he introduced me to modern statistical mechanics. And after that, I was lucky enough to get a tenure track position here, which meant that the dreaded clock started ticking. Then my wife, Elizabeth, in the audience, she joined BU as a senior research scientist. And she has been a very, very strong supporter of my career, so that is fantastic. And we have collaborated so much, and she deepened my understanding as well as my curiosity about fundamental biological questions. Lastly, I must mention Ramakrishnan, also in the audience from Harvard, because he changed my thinking about entrepreneurship. And so as a consequence, in 2019, we formed a company called Mechanobiologics. And then these are, I also wanted to mention a few of the lab members. I formed an independent lab in the middle of the 1990s, and I was lucky to get postdocs from all over the world, from France, Italy, Spain, a lot of physics students and postdocs from Brazil, as well as MD PhDs from Japan. And this is just one of those lab dinners after lab meeting. This is here Steve Harold. He's now a big boss at United Therapeutics. And then this is Hiro Sakai, an MD PhD from Japan. That's Kelly Brewer, a PhD student at the time. Steph Kononov, she was a master's student. Right now, she's a leader in a local engineering company. This is Adriano, a physicist from Brazil. He's now actually a full professor of physics in Brazil. And this is Elizabeth, and this is Sinei. He was a superb, talented, double major BME student, worked in the lab for more than a year. This is about 20 years later. We look the same, by the way. This is Caleb Alves. He's a physicist, a physics PhD student from Brazil. That's Adam. He's also here. He was a systems engineering, and he's now a pulmonary physician. And Jared, he was an MD PhD student, and he's now a thoracic surgeon. And then Kohei, he was a Japanese medical student. Samir, also here. And Yasmin, also here. So good to see you guys. And these were all fantastic PhD students. Then we used to have barbecue parties also in the lab, and this is just to show you a couple of other people, like Lauren Black. He's an associate professor at Tufts at the moment, and this is Apirade. He was also talented experimentalist. And this is Harry. Harry, where are you, Harry? He's over there. He's an associate professor at Northeastern, and this guy is a weirdo. But he was a terribly talented physicist. And then this is almost 20 years later again. You can see Samir, Jared, and a bunch of undergraduate students. And then we went to conferences, and then finally, these are current PhD students in the lab, Joseph and Celeste. All right, so this slide is difficult. And the reason is that I went to the lab a couple of years ago just to clean up, and then I found a note, like a cartoon, in the lab. And it was done about maybe 20 years ago by somebody in the lab. I don't know who the person was. I have some guesses, though. But I debated whether I should show you or not, but this is it. All right, so the title is The Lab Food Chain. And you can see that an invisible hand from the sky instructs an angel to carry a bag of cash to a faculty. So, but it looks like it's me. And then Steve, this is on the first floor. Steve is also on the first floor, maybe even on the floor. And then he instructs the graduate students who are in the basement, working very hard. And then you can see the master student is in some sort of a cave. And these were undergrads. I don't know. And fortunately, the undergrads finally actually made it to the cave level. So anyway, I stop here with the introduction, and let's move on to the first topic, biomechanics. So around 1999, a student, Steph, I already mentioned her, joined my lab as a senior, and her job was to design a stress strain measuring device. This was one of the first devices that could actually visualize microscopic structure while measuring the macroscopic stress strain properties. And in fact, I know that it was the very first one in the lung community. So you can see here a lever arm, a wire here. This is the sample, and then the wire. There is a little plate here that was glued to the tissue, and another plate that was attached to a force transducer, and then you can measure the stress strain curve. This is the tissue bus, and then you can put the whole thing on top of a microscope. And that was good. We started out looking at things like stress relaxation in the tissue, but this did not lead anywhere. So I asked one of my Japanese MDs to create an animal model of emphysema, which is how actually the emphysema project started in the lab. And he created the animal model and then gave some of the tissues to Steph. She measured the properties, she imaged under the microscope, and then one day she comes to the lab and she says, Dr. Suki, there is a problem. The tissue ruptured. And I asked, what tissue? Well, it was the emphysematous tissue. Did the other tissue, also the normal control tissue, rupture? No. So that was interesting. Did you take images? Yes. So we went back to the lab, and then we tracked down all the images that she had, and this is what she found. So this is a collagen-labeled tissue and of an emphysematous lung. These would be the alveoli. These arrows mark an anatomic landmark, and this was under a uniaxial strain of 10% in the vertical direction. Now, when she stretched it to 20%, well, these landmarks actually disappeared. And then when she stretched it even further, the tissue ruptured somewhere around here. So that's actually evidence that the tissue from the diseased lung can rupture under mechanical strains. And these strains can occur during breathing. And that was a major discovery at the time. And to elaborate on this, let me give you a short introduction to emphysema. So it is actually a subset of COPD. And COPD is chronic obstructive pulmonary disease, number 4 killing disease in the world. And COPD, the primary risk is cigarette smoke, and it cannot be cured at the moment. Now, what happens is in emphysema is that basically what people find is that these alveolar regions get larger. It's called the airspace enlargement. But at the time, it was not known how. So rupture is the mechanism how this can happen. But does it actually happen in humans? So about 25 years later, Celeste took images of human lung tissue slices from a COPD donor. And this is a multiphoton image. The cyan is actually a second harmonic. And what we saw here is this little thing here. That was very curious. It's kind of like the end of the wall. So that was very interesting. And hence, we looked in the confocal Z stack whether it's just that piece of tissue went out of the focal plane, but we didn't find it. So when we zoom in, this is what we see. And what you can see here is really a very highly wavy fiber. And the second harmonic labels, actually not labels, but images the type 1 collagen. So this is actually evidence that there is rupture. Why? Because the tissue is under stretch, and when something ruptures, the retraction of the tissue actually curls up the collagen. We don't see this in normal tissue, therefore this is really evidence of tissue rupture in human COPD. All right, so how do we go on? Well, we follow two directions. One of them is to try to understand why rupture occurs in the treated tissue, and the other direction was to try to understand the implications. So looking at the first one, this is the the stiffness of a piece of a normal tissue over a period of 60 seconds. And that's how the tissue looks like, very nice and homogeneous. Now we put enzyme into the tissue, elastase. Now elastase, in this case, it's porcine pancreatic elastase, but elastase is known to digest elastin. And the elastase itself is also known to be involved in COPD and emphysema. So what we can see here is that the stiffness decreases over time, and consequently, the structure is slightly different. Now, then we repeated the measurement under a mechanical tension. And we applied a static 40% strain on the tissue and added the elastase and monitored the stiffness. And you can see very significantly higher decrease of the stiffness that resulted in a very, very destructed tissue structure. So, but the question is, what is the mechanism? To do that, we turn to fluorescence recovery after photobleaching, which is that we need fluorescent particles in the tissue and then see how it recovers after bleaching. So Elizabeth labeled elastase particles with fluorescent tags. This is a red fluorescence. And then basically when the fluorescent elastase is put in the solution, it actually binds to the tissue. And that's why you see it red. And then we find a place shown here by the white circle, and then apply a strong laser power so that we can photobleach the tissue, the fluorescence there. So you can see that the fluorescence actually disappears. And then just wait and take images over time to see that this actually recovers. And the reason for the recovery is that the unbleached elastase, red elastase, will diffuse into this area, and then it will again bind to the tissue. It turns out that the recovery process is fairly nice. It's an exponential. And when we fit an exponential to the curve, then we get a time constant of 83 seconds. And when we repeated this in a tissue under tension, the recovery was faster, and it resulted in a time constant of 37 seconds. Using a mathematical model, it is possible to show that the inverse of this time constant is actually the dissociation rate constant for the chemical reaction between elastase and elastin. And then we could construct a diagram shown here of the dissociation rate constant or binding off rate as a function of the microstrain that we estimated from these images. So that's very interesting, but what about collagen? So Elizabeth also developed a method to isolate a single collagen fiber from the lung tissue, and Celeste used this, and you can see a single collagen under the scanning electron microscopy here. And then if you zoom in, what we can see is that the fiber is composed of many wavy fibrils. Then another postdoc, Jake Herman in the lab, he worked with Michael Smith, and Michael Smith allowed him to work in his lab and basically helped him to construct a single fiber stretcher system. And that's shown here. The fiber, the collagen fiber is here, I don't know if you can see this, but these are two glass rods. One of them is used to stretch the fiber. The other is, the bending of the other is used to calculate the force. And that was done, and Celeste measured the stress strain curve of a single fiber under the influence of collagenases. Now, those are enzymes also involved in COPD, and what we see is that the straight region here, the slope here is the stiffness. It goes from 80 megapascal all the way down to 13. And then she also looked at things like the yield stress. Now, the yield stress is basically the stress in the fiber at which or beyond which single molecules start to slip on each other, and then fibers, the fibrils start to slip on each other, and so forth. So it's a multi-scale problem, and you can see that the digestion actually significantly lowered the yield stress. So to sum up this part, I would say that essentially we found some mechanism related to how mechanics and enzyme chemistry interact in the tissue. And one of the main conclusions is that this is not like a regular chemical reaction as that would occur in solution. Instead, mechanical forces have a huge influence on how enzyme works. So the prediction from this would be that in places in the emphysematous lung, when there is inflammation produces a lot of enzymes, elastase and collagenases, mechanical stresses there would rupture. The higher that stress, it would rupture the tissue. So next to then the second topic, fluctuations. If you start inflating a lung of for example, a normal person or an animal, that what you see is that as the volume goes in, the pressure nicely, continuously increases, nothing fancy. But if you have a region, or maybe the whole lung that is collapsed, then that could be represented by this. These thin lines represent closed airway segments and closed alveoli down here. So in that case, it turns out that each segment, airway segment, has a critical opening pressure. So when the pressure across that segment reaches in the lung, reaches the critical opening pressure of that segment will open. That will generate a sound called a crackle. And that's kind of like when you open a bottle of champagne, that generates a sound. So this is like a tiny champagne in the lung. But then the pressure goes up and down, and the part that goes down reaches the next segment. And I will show you this. So maybe this opened. And then if the pressure wave has an amplitude that is already larger than the critical opening pressure of the second guy, then that also opens together with the first. And this is so fast, actually, that we call this an avalanche. And the avalanche stops when the pressure here is lower than the opening pressures of these guys. And then, of course, this process goes on as you inflate the lung. And then some of these avalanches can reach the bottom of the tree and opening alveoli. Then air, through this path, rushes into the lung and fills up the alveoli. And then eventually, you can fill up the whole lung. But this occurs in big steps. And therefore, the transition of the lung from the closed to the open state is kind of like a phase transition. It's very rapid. We did this in collaboration with Gene Stanley's group. We published this in Nature, and I followed up with a lot of interesting papers that I won't talk about. But the question is, does it have, this was a new concept that people did not know and did not appreciate at the time, but does it have any clinical implication? And it seems like it does. And for that, let me just briefly introduce a paper from the literature. A group from Canada, Lefebvre and colleagues, argued that when we ventilate a lung, then the tidal volume that the lung receives in the ICU is always the same, just back and forth, always the same amplitude, same rate. But we don't breathe like that. Therefore, what if we actually replace this monotonous breathing with a variable pattern that is kind of simulating or mimicking natural breathing? They did that in an injury model in pigs, and then this would be their variable ventilation. This is arterial oxygen tension, and this is the so-called conventional ventilation. And they found improvement, but there was no explanation. Why does it occur? At that point, we thought that maybe we could explain this with the avalanche model. So we came up with a model to do that. And this is the pressure volume curve that I mentioned to you. This is a very sharp transition here. And then when you ventilate the lung, say, between P1 and P2, you always go along this curve, and then you open the lung up to V2. Now imagine that we add a Gaussian distribution on top of P2. Now you ventilate from P1 to a little lower than P2, you lose volume. But then the next breath maybe is above P2, and then you gain a lot of volume. And over many, many cycles, the average P2 is the same. However, the average V is now not V2, but V3. And this can be as big as 200% improvement. And then we also predicted that actually, if you change the standard deviation of this Gaussian, then you can predict that there must be an optimum level of variability that maximizes gas exchange. So we teamed up with other people, and then we published a short communication in Nature. Now, I actually display here the entire authorship because this was a powerhouse paper, including Ken, he was the chair at the time, Jim Collins, a superstar, Jose Andrade, also a very famous physicist from Brazil, Edin Genito, a very well-known MD from the Brigham, and then Gene Stanley. And that was, the paper got a lot of attention, media attention. And even there was a short article in the New York Times, and we were so happy. But then one day I got a call from California. A guy called me up and then said that he read the paper, and his wife is in the ICU undergoing mechanical ventilation, and the doctor said that she doesn't have a lot of chance to survive. Could you help me? And I felt so bad because I couldn't. And but that story motivated us to go on and then test this further experimentally. Then Steve Arold, I already mentioned, he was an undergrad in the lab back in around 1999, and he was doing his senior project, and told him that, okay, here is a mechanical ventilator, program it to deliver this ventilation whereby the volume delivered changes from cycle to cycle. So the black shows you the conventional ventilation. Still, actually, this is the current intensive care ventilation protocol. And the red is the variable ventilation pattern. And then he used endotoxin-treated guinea pigs, and this is what he found in one of the guinea pigs. The population is slightly different, but this is just the typical best result. I just to remind you, this is what we predicted 4 years ahead. So we thought that this is fantastic, so we went on, we got grants to do studies, and I would just like to show you 2 studies briefly. One from Apirade. She treated mice with hydrochloric acid to generate a typical ARDS, or acute lung injury model. And this is the alveolar structure in a normal mouse. This is when the injured lung was ventilated with the conventional ventilation. You can see a lot of collapse and heterogeneity. And this is when the lung actually was ventilated after 1 hour of the injured lung. Look, this is almost as good as the normal that was not even injured. So that kind of a structural difference showed up in function. Now these are peak pressures, and then during conventional, again, this is the standard ICU protocol. During conventional ventilation, it goes up straight linearly with time, but it plateaus during our variable ventilation. To look at the effect more carefully, we did a lot of studies, including, and I won't go into this, but these are all percent improvement of variable ventilation over the conventional. And For example, gas exchange, mechanics, even surfactant proteins, and then EC adherence. These are the proteins that link epithelial cells. And this was obtained from lavage fluid. But these proteins are not supposed to be there in the lavage fluid. If they are there, it's a direct epithelial injury marker. And also IL-1 beta, a inflammatory marker. And lastly, among many studies, we collaborated with a group in Australia, and they had a setup for doing measurements in preterm lambs. And the results were really similar to what we obtained in mice. But one interesting finding was this. We also measured cerebral tissue oxygen saturation. And the conventional ventilation had a value of 72 or so, and this is highly significant during variable ventilation. So if a preterm baby needs mechanical ventilation, and they do, unfortunately, occasionally, then this variable ventilation actually could be and should be useful in terms of brain development. So I think that the implication is very interesting. All right, so additional studies. We patented the method and then did a small clinical trial. And eventually, with some air, we published a paper in Scientific Report that showed improvement in respiratory physiology using the variable ventilation method. And then I'm really excited to announce that we actually got just recently, like a month ago, a $8 million DoD grant, a multi-center clinical trial to test variable ventilation in human subjects, so human patients with acute lung injury. And as of today, 4 p.m., DoD has not terminated this grant. So the question next is, but what is the mechanism at the cellular level? And then we can roll back the clock up to about 2003. Steve Harold, when he finished the animal studies, he said that, well, I would like to stay in the lab for a PhD. Okay, good. And then his PhD was to design a cell stretcher, shown here, and then to test the variable stretch mechanism on type 2 epithelial cells, which are the cells that produce surfactant. And so he labeled surfactant lipids with radioactive probes, tritium, and then he treated the cells in culture with that. The cells take up the radioactive tritium, and then they incorporate into the phospholipids, and then later on they actually secrete radioactive lipids. And so what you see here is the phospholipids secreted by the cells when they were ventilated for 1 hour without variability. So that follows the conventional mechanical ventilation. So that's about 1.2%. Then this is the next point. And so 2.5% variability in the peak stretch did nothing. Steve was really disappointed. He was very close to a nervous breakdown at that point. But fortunately, when he increased the variability to 5%, then lipid secretion actually doubled. And then these are the rest of the data. It seems like maybe there is an optimum level of variability. So that's actually very, very interesting. So at that point, once we published this paper, I was thinking that this phenomenon whereby stretching with a fluctuation in the peak amplitude cannot be specific to lung cells. And I hypothesized the following. So cells eukaryotic cells started to form multicellular organisms about 1 billion years ago. And, but they were always exposed to different fluctuations, mechanical and all sorts of things, chemical. And about 600 million years ago, complex organisms start to evolve. And these organisms already had a closed circulatory system. And then during their just life, there must have been a lot of variations in blood pressure, heart rate, breathing rate, and muscle contraction, movement. And so all the cells in the body that are sensitive to mechanical signaling, they must have been exposed to this. Therefore, evolution must have built this kind of a fluctuation into cell function. And to look at one of the most ancient signaling mechanisms, we decided to actually probe ATP production. And here is how. We took vascular smooth muscle cells in culture first and exposed them to 3 different protocols. Unstretched, that's the blue. This is a strain amplitude. And then the monotonous stretch, which is the black, or the red, which is the variable stretch, directly applied to the cells. Now, the maximum as well as the variation around the maximum were kind of tuned, actually it was calculated to be relevant for blood pressure variation in normal human subjects. And then we used a dye called TMRM, which collects on the inner membrane of the mitochondria. Now, this means that it is actually related to the mitochondrial membrane potential. And so if the dye intensity is higher, then what happens is that there is a higher voltage on the mitochondrial membrane. Higher voltage, in turn, will generate, will send down more protons down the ATP synthase, and that will result in more ATP production. So this is how it looks like. This is a tubular structure in the unstretched group. And that's after monotonous stretch. You can see that the network actually is denser and the intensity is higher. But what happens next is when you add the blood pressure variability related stretch fluctuations, that's it. This is a super dense network, very high intensity. So then we looked at about 1,000 cells, and this is the result, very significant. So it seems like, I call it ATP production. It's not really, but it's just more or less intensity. But nevertheless, it is related to voltage and hence ATP production. But there is a huge difference between the monotonous stretch and the variable stretch. Then we wanted to look at whether it is a cell phenomenon or maybe this also exists at the level of the tissue. And then came Yasmin. He measured really carefully the contraction of vascular smooth muscle, vascular tissues, like a piece of aorta in a stretcher system, mimicking the variabilities due to blood pressure. And what he found was that 4 hours of monotonous stretch, which is actually the standard laboratory condition, downregulated contractility, but the variable stretch actually maintained contractility. So clearly, 2 conclusions from this. One is that there is an effect of modulating ATP production on other cell functions, and this is not related to cells in culture, but it's also at the level of the tissue. Then Elizabeth did another beautiful study, whereby we were interested in whether now, is it possible that different amounts of variability will have different effects? And she labeled mitochondrial ROS production, and we used 4 different levels of variability around the mean. V0 is the same as the monotonous stretch, so 0% variability. 6% variability, V25 is the same as the variable stretch in this group. And V50 was selected on the basis of what happens in, for example, cardiovascular diseases. It is known that blood pressure variability goes up. It's an independent predictor of a lot of cardiovascular problems. Not only that, but it's actually also a predictor of cognitive decline. It's very surprising. In any case, another interesting fact is that blood pressure variability goes up with aging. So then the conclusion is this, or maybe a speculation. ROS, especially mitochondrial ROS, is known to be involved in aging. Really, apparently that's one of the key drivers of aging. And, but that increased variability in blood pressure actually drives off the reactive oxygen species. So perhaps it has something to do with aging as well. What are the mechanisms? This is a schematics of the mitochondria within the cell, and I'm not going to talk about it a lot, but just to say that this is the ATP production from the previous slide. So this is the control, and then we wanted to prove different aspects of the relationship between, for example, actin and mitochondria, microtubules and mitochondria, just to figure out the signaling pathway. And Elizabeth did this, so first, when the polymerization of actin was disabled using cytokinin D, there was no effect. So that's actually rather interesting, in fact, unexpected. When we inhibited the polymerization of microtubules, it actually dissipated the difference between fluctuations and the monotonal stretch. So then, of course, she used another 13 different inhibitors, a total of 15, and I can't tell you all of the details, but the following really complex picture emerged. So this is the kind of biologist type of a signal transduction pathway, except my addition to this would be structural networks. So the left is what we call classical mechanotransduction, and the right is the fluctuation-driven mechanotransduction. And then without going into the details again, but what we found is that basically the network, a mitochondrial network, is much denser, and that results in an increased ATP production in the fluctuation-driven mechanotransduction. And in turn, this reduced laboratory standard monotonal stretch reduces cell activity, but it actually maintains or upregulates in the case of variable stretch. So I wanted to tell you a quick story, how we published the paper. I was so excited when we got some of these results that I thought that maybe this is so interesting and so important, and it has so wide implications from mechanotransduction that it should actually revolutionize the field of mechanobiology. But I was cooled down very much, very soon, after the paper was rejected from basically every possible major journals. And until finally, after much struggle, we published it in Nature Materials. But I still think it's an important topic. Because any time a cell function is sensitive to mechanics, it should be sensitive to slightly larger and slightly smaller amplitudes. And so, but despite of this, virtually all labs in the world still uses the good old sine waves with the same amplitude. And despite of the fact that basically our cells in the body have never seen, sinusoids over the 1 billion years of evolution. So topic 3, this is going to be shorter. I wanted to start with a quick quote from the British-born American statistician. He said that all models are wrong, but some are useful. So over the decades, we actually carried out a lot of different modeling studies. I listed some of the contributors here. I don't want to go into this, but just to show you the breadth of different topics. And at the end, my conclusion is that we actually need to take George Box's statement to the limit. And I would say that the ultimate usefulness of a model is when it is replaced by an even more useful one. So, and that will come up soon. I wanted to actually show you one modeling study back from 2002 that I did myself. This is, again, remember, Steph did these studies, rupture of alveolar walls. And so what are the consequences? And I just created a simple-minded elastic network of springs and cutting a certain fraction of the springs that I call chemical, because if this was like enzymatic reactions in a solution, then you would expect this would be random. And I used to ask the audience that, what do you think? How many springs are missing from this network here? And then the answers would range from 5 to 20%. The actual number is 13%. Then this is a totally deterministic model, what we call a force-based model. In this case, we initially cut a few. We solve the network for equilibrium, find the springs that carry the largest force, and then cut those. So it's really deterministic. Then I would ask the audience, so what do you think? How many springs are missing from here? And then the answers would be between 30 and 80%. But the actual number is 13%, the same exact as up here. And that's really surprising to many, because it is just that the method of cutting has a huge influence on the actual structure. So when chemistry is influenced by mechanics, I think the bottom one should be more relevant to basic physiology and pathology. How do we compare this to real life things? And I took a CT image that we published back in '99. This is a real CT image of a person with emphysema. The colors code low attenuation areas where you could think of those low attenuation areas where the lung tissue already ruptured. And so the different colors mean nothing. It's just to label different clusters of low attenuation area. Then we can measure the distribution of those clusters, and it looks like this. On a double logarithmic graph, it's a favorite thing. It's a power law. It's a fractal. Now, in this model, we said that, well, let's use basically every little things here where there is no spring and connected air spaces as a low attenuation cluster. And then we can calculate the cluster size distribution. And that looks like this. And it is just not like the real one. And then, of course, we did this for the force-based, and that looks like that. So that's a great agreement with the data. And I would say that the simple network model can bridge the gap from basic science of rupturing the walls to clinical things like CT images. And I would also say that this model has reached its maximum usefulness because some of my students and postdocs have replaced it with better ones. And for example, Harry did a really very nice study. He extended this to 3D and then used various cutting mechanisms and then linked this to functional decline. And then we also looked at fibrosis, pulmonary fibrosis. And especially there is an interesting structure here. The red things are low attenuation, and the cyan are thick collagen deposition in pulmonary fibrosis. And if you zoom in, what you see here is that the red surrounded by strong collagen, and this is called the honeycomb structure, the subpleural honeycomb structure. How do you explain that? Well, Tyler Wellman, a postdoc in the lab, also BU graduate, came up with a cellular model. He added agents on top of the network models, and then he got this, and then the statistics of these clusters agree very well with the statistics of these. So that's actually pretty nice. Moving on, Joseph, current PhD student, he further improved on this model. What you see here is the histologic structure of pulmonary fibrosis. And for examples, if you zoom into these circled areas, then we get these structures. And Joseph's model actually recreated the following. This is, and we published that last year, but I think the correspondence, even if statistical, is really, really amazing. What is nice about the model is that it links now self-healing with fibrotic pathways at the level of cell signaling. So then my last topic is technology. So I talked about variable ventilation. We did a lot of other things, designing various stretchers, a bracelet for infants. But I just wanted to mention one technology development, what we call the Mechanovel. The purpose of the Mechanovel is actually to measure stiffness of thin tissue slices. And this is a joint invention with Ramakrishnan's group at Harvard. And this is an actual picture. And if we zoom in, what we see is that at the bottom we have a membrane, and then this membrane sits on top of an indenter, and the sample is on top of the membrane. When you rotate this rim, then it moves down like this, and it pulls the membrane, and together with the membrane, the sample is stretched as well. So if we zoom into this part here, then it turns out that the membrane is actually a composite material consisting of a silicone membrane and a soft gel with impregnated fluorescent beads. So what we do is that we stretch this composite with and without the sample. We detect the displacement field from using a microscope, and then we can calculate the strain field over or under the sample, and from this we can back out, we can calculate the stiffness. We are working on actually a multi-well version of this, and so the stiffness would be available per well. And we patented this, and the plan is that Mechanobiologics will actually buy the license and then eventually produce and sell this multi-well stiffness measuring device. We think that this should be very useful in drug development. Why? Because when you look at drug developments in cell culture or even animals, basically it's very, very difficult to get a functional readout. And then here is how we can some functional readout in the Mechanobiology. This is the stretching of the sample. This is the actual beads after the stretch. And we used, again, human thin lung slices from healthy as well as COPD donors. And these two images show you the strain field. And then this would be the stiffness from the healthy, and this is the stiffness from the COPD samples. So COPD downregulates the stiffness, but we know that. It's nothing new, because we found rupture and we found the elastase induced basically softening of the walls. So which one is it really? What is the dominant mechanism? And so Joseph used a kind of a personalized network model. And then based on that, again, no details here, but he was able to estimate the stiffness of the single walls. And that is the important thing. And what we can see is that in COPD, the stiffness of the wall drops to half of the healthy case. Now, lastly, just recently, we had a really new development done by Jae-Hoon Kim, also in the audience. He used to be a senior scientist in my lab, but now he mostly works for the company. And then he came up with an estimation method to get the stiffness map out of these images. And this is actually, this looks kind of like with the stiffness map that you can get from an AFM. However, it has its own advantage, so it is actually better. I won't say that, but it is more suitable to measure the stiffness map for tissues that have like the lung cavities, because you can't really probe with the AFM those cavities very well. So that's all I have, and I would like to just thank everyone, and especially I'm grateful to my mentors and collaborators within the department, obviously Ken, Dimitri, Hari, as well as Michael Smith, even Joyce at some point back. And also outside the department, I collaborated a lot with Catherine Zhang and now with Bob Varela. These were all fantastic collaborations. I also thank my postdocs from all over the world, the PhD students, master students, and undergraduate students, and my family, my parents, Elizabeth, my wife, my two sons, and recently new additions to the family, 2 grandkids. Thank you so much.
We have time for questions.
Hi, very interesting talk. I was wondering if you had ever looked at any COVID-19 lung damage, or how you think would be a good way to study that.
Well, fortunately, COVID-19 is over, so we don't care. But yes, I think COVID-19 is really interesting from the point of view of that it very often it leads to pulmonary fibrosis. And I actually have been involved in some studies where a patient had a lung function test and CT images that looked worse than the most severe pulmonary fibrosis conditions. Yet, the patient recovered. If you have pulmonary fibrosis, like idiopathic pulmonary, you cannot possibly recover. And we just don't understand why. In one case, you get fibrosis from COVID that can be healed, but the regular pulmonary fibrosis does not, you cannot recover from that. So I think it's an open field. It would be useful to study, because maybe studying this, you can better understand also idiopathic pulmonary fibrosis.
I actually don't have a question, but I want to say thank you so much. It was amazing to watch your presentation, and it was really an honor to be part of the lab. And this is, I think you should also deserve an award for being a great mentor, because you influenced so many of us, so not only contributed to scientific, but also to great mentorship. And I'm proud to have been part of your lab. Thank you so much.
Yeah, thank you. Yes.
Yeah, that was great, Béla. So can you talk a little bit more about the clinical trial? That seems really very, oh, sorry. That was really wonderful. Can you tell us more about the clinical trial? Just in terms of what that's doing, I certainly appreciate what you're saying about the variability and how we're still doing things kind of uniformly. Can you maybe tell us more about what the details on this multi-center trial?
Sure. Basically, this did not come easily either. We submitted several times. We got very close, and finally this year, it got funded. 6 centers are involved: Bay State Health in Massachusetts, the Brigham, one in Oregon, one in Alabama, one in Colorado, and one in Arizona. And the idea is that we will set up the things here at BU first. A PhD student and me, we will actually try to develop a computational model of the acute lung injury, and then come up with the actual protocols, and then it goes through all the doctors, and then these centers will just do the studies. So the first part would be like a 2-year in which it's more like safety, followed by another adding like 200 something patients, whereby we would go for, if the first part is successful, we would go for, is there a benefit? If it's safe, then is there a benefit? So this is how it's set up.
So is it tailored is it customized for each patient?
No, that's a great question. No, not at the moment. We could do that, but it's just simpler than that for now. Yep.
Perhaps a variation on the first question. The variable ventilation results, a lot of the key results are 10 to 20 years before COVID, or 5 years after COVID. And it was kind of disastrous what happened during COVID with the ventilation. Could anything have been obviously done differently?
I would think yes, because it turns out that, as far as I know the statistics, if somebody had to go into the ICU, and then the person had to, doctors decided that the person had to undergo mechanical ventilation, the chances of survival actually dropped. So somehow the mechanical ventilation was not done carefully. And I can, in fact, let me tell you a quick story. With a doctor in Hungary, we published a paper. Again, this was very difficult, because that's the patient that I mentioned before. And he said that we can't use the ARDS protocol, which is kind of high lung extension called the PEEP, positive end expiratory pressure, and then small amplitude ventilation to sort of protect the lung. Why? Because the pressure volume curve is very flat. It's a fibrotic lung. Rather, we have to lower the PEEP and then add the large tidal volume to get enough air into the lungs. Because if you ventilate right here, you can generate a huge pressure, but what you get is like 50 or 100 ml into the lung. But if you lower the PEEP, then you go this way on the pressure volume curve, and then you can get 400 mLs. And apparently, this doctor saved a life with this change in protocol. And then when we submitted this short communication to journals, well, they said, well, this is great, but it's not the established protocol. So it was rejected. So I think there is plenty of room for improvement.
Wonderful talk. Thank you. I've got a question about the variable ventilation and the signal pathway slide that you showed. Can you comment if you've measured effects on the immune compartment in the tissue?
Okay, that's an interesting question. Well, in one study with prematurely born lambs, we looked at a bunch of proteins from the lavage fluid. For example, surfactant protein A, B, C, D. Mostly Elizabeth did the work. And so by B and C, they are useful for the surface tension of the lung. A and D, they have some sort of an immune function. And they were also improved by the variable ventilation. I do not have any, I don't know how, but I think, again, this has to be related to the different patterns of stretching of the alveolar cells will generate different signal transduction pathways. And ultimately, this will lead to different expressions of proteins. But I don't know exactly how.
Great talk, Bela.
Thank you.
I'm thinking about the variable ventilation as well. You showed one type of variation on the sine wave, but there's an infinite number of variations you can look at.
Yep.
Have you tried, for example, constant amplitude but variations in frequency, or changes in amplitude but no changes in frequency, or other things like this to figure out what really matters?
Okay, so that's a great question. So what we decided to, so if you have a large tidal volume, we wanted to have the same ventilation of the lung. So what we did was that large tidal volume was associated with a very long time period, so a low frequency. Therefore, the product of the two, which is ventilation, was kept constant, so as to not bias oxygen partial pressures. But we did try different amplitudes, but we also always scaled the frequency. And as some of the slides showed that, in fact, there seems to be an optimum level of variation in amplitude. I know some groups tried no change in frequency and just amplitude. But that, to me, it's like you change two things at the same time, because you change minute ventilation and you change the amplitude. So it's hard to compare the different groups. I think there is a literature on, probably you know this better, but if you change the rate significantly on how the cells are stretched, the higher the rate, you get more injury. And so we just didn't want to do large amplitude, very high rate.
Thank you, Bela. It was good. Can elastin inhibitors have a favorable impact?
I'm sorry?
Elastin inhibitors can affect lung behavior.
Elastin inhibitors. Elastase, maybe?
Elastase, sorry.
Yes, I think there is a genetic version of emphysema, the alpha-1 antitrypsin deficiency. And I believe even probably Daryl Cotton at the medical school, they used something like an antibody-related, don't know the details, but I think this has been tried. Still, I don't think there is because when you smoke, the inflammation will generate a spectrum of enzymes. And if you just pick one, your favorite, you might, attenuate something, but there is plenty of other things that will happen. So my personal opinion is it's not going to work.
Okay, we're going to wrap up the formal part of the program here. And certainly people can mill around and ask questions of each other, ask questions of Bela after this. But let's give another round of applause.